Hey, Bio 1 students, Mr. Yance again. We're going to continue on with a little mitosis lesson today. We have, in previous notes, talked a little bit about interphase, which is not a dividing phase, G1S and G2, as you can see up there. I covered that the other day. And that's where the cell spends most of its life and where cell grows, matures, and does cell stuff. Today, we're going to talk about this slice of the pie. Prophase, metaphase, anaphase, telophase, and mainly talk about mitosis and how it's different than these other phases, okay? So let's just jump right in. So we're going to talk about prophase. Prophase is the first dividing phase. That's why we call it prophase. You can see that on my screen. Pro means first, and it's well named because it is the first cell division phase. And we divide it into an early and a late. Some textbooks, some diagrams may call part of this pro-metaphase. I kind of like to use the words early and late. And when the biggest thing, and you notice that from the lab that we did and looking at pictures, color sheets and pictures on the internet and WebQuest and things like that, that there are certain things you got to look for to know you're in prophase. So how do I know I'm in prophase? What you're going to see is the nucleus and nucleolus are going to start to disappear in early prophase. They may still be present, but they're fading. Centrioles move to opposite poles. So in the cell, think of the globe. We have the center of the globe. That would be the equator and the poles, north and south pole. In this case, we're going to have an east and west pole. We use the same terminology with cell division. And if you guys remember from cell chapter, we talked about astral rays. These little fingers come out from the centrioles and they start to go towards each other from either side, and that's called an aster. And the biggest thing you're going to look for is chromosomes will appear, okay? So when you look at, here's a diagram, you see the nucleus, nucleolus may still be there in early prophase. These centrioles are starting to move. There's those little astral rays. The whole thing's called an aster. Okay, and you see these little gummy worm things show up. That is chromosomes. Okay, and that's the biggest sign that you're in early prophase, as these chromosomes appear for the first time. So that's a great picture right there of an animal cell, and you see that the chromatin, which was that unorganized mess of DNA you see in interphase, it's going to start to get organized. You're going to see these little worm-like gummy things show up. I like to call them gummy worms. And that's how you know you're in early prophase, okay? And you can still see a little faint nucleolus still in the background, so that's how I know I'm in early prophase, okay? Now, it's going to progress pretty fast into late prophase. Just a little terminology you need to know. A chromosome is the whole thing here. It looks like an X. One leg of it is called a chromatid. Remember that? That's a single strand. There's two of them hooked together. And a centromere is this narrow waist that is the center part of a chromosome. So kind of remember those terms. We're going to come back to them here today when we go through these different phases. Now, in late prophase, the only thing that's different than early prophase is the nucleus and nucleolus. They just are fully gone now. We have spindles formed. The centrioles are across from each other, and the astral rays unite and form these spindles. And sort of like hanging clothes on a line, these chromosomes at the centromere here in the middle, they're going to get tethered to that spindle. So you can see these little gummy worms starting to get organized. And this is late prophase. The centrioles are getting a little closer to the poles. They show a dotted line. You don't see the nucleus, nucleolus anymore. It's gone. And now I'm ready to progress into the next phase. Okay, so really, early and late prophase are setting up metaphase, which is the next phase. So everything's getting in line, everything's getting hooked up, and we're going to progress into a very brief and quick phase called metaphase. All right, hopefully that makes sense so far. Okay, meta is a really fast one. It's easy to talk about. Meta means middle. So these chromosomes line up at the equator in the center of the cell, and they attach they're attached to the spindles. I'll show you a picture in a minute. And that's how you know you're in metaphase. Hard phase to find under the microscope because it's so brief and the cell doesn't spend a ton of time in metaphase. But it is a very distinct, easy phase to see. There's just not a lot of, see if I can get clicked on here. There's just not a lot of, let me back up just for a second. So hopefully, there we go. So there's metaphase. It almost looks like someone's taken a marker and marked in the middle of the slide. If you look really close, you see the spindles attached. You see these chromosomes. I mean, they're just lying one on top of the other. Good diagram over here. Now we're going to have a little tug of war. Okay. Now remember, in mitosis, we want to keep the chromosome number the same. Okay. Now to do that, this is where it gets tricky. So let's use this illustration. I have 1, 2, 3, 4 chromosomes lined up. I want to end up with 4 when I'm done. So watch what happens in anaphase. So in anaphase, we get this tug of war occurring, and we pull these chromosomes apart. Not keeping them together, we pull the chromatids, one leg of a chromosome, we pull them apart. And that way, I keep the chromosome number the same. If you look here, 1, 2, 3, 4, 1, 2, 3, 4. Remember in interphase, during S phase, here it is, we made a copy of DNA. So that means I have one copy going this way, the identical copy going the other. Now I got the same information in both cells. Under a microscope, it's easy to see because you can see them pulling apart here. And that's how you know you're in anaphase. Okay? Now I am ready. So now, once those chromatids get all the way across to the poles, now I can divide the cell. So telophase means the end, another great name. The cell is going to pinch inward in an animal cell. In a plant cell, we have a cell plate that comes down, and we are going to divide that cell into 2 daughter cells, but they have the same information. They have the same chromosome number. You can see this happening in these 2 slides. Now my mitosis is complete. I have the same information, and the only thing I've done is increase the number of body cells, skin, blood, muscle, bone, that type of thing. Okay? Now, don't get confused because cytokinesis is not a part. It is a part of mitosis, but it's not a phase. It's not a dividing phase. It's like interphase. Cytokinesis is how telophase splits the cell. It's just a process. So in animal cells, we have a cleavage furrow. Plant cells, they have a cell wall, so we have to have a cell plate. That's all cytokinesis is. Don't get confused with that. I think sometimes students think, oh, that's a dividing phase. It's not. It just completes the process of cell division. All right. Now look what happens. Very important point. What I'm going to end with today. Okay. Look what comes back. The nucleus comes back. The nucleolus comes back. The chromosomes turn back to chromatin. What am I back to? Interphase. It is a cycle, right? So basically, if you go back and you look at, let me go back a second here. If you go back to this picture here, right, what we started with, a cycle means I start and stop back at the same point, correct? So you can see I went through prophase, metaphase, anaphase, telophase. There's my dividing phases. Cytokinesis is going to split that cell into 2 daughter cells, down back to G1. The difference is I have 2 cells now instead of 1. Okay? So I hope that makes sense. We're going to progress into meiosis in the next set of lecture notes. I'm going to get all these lecture notes online so that you guys, you virtual students, are all caught up with my traditional classes. And we'll have a test coming up here pretty quickly. So, we'll wrap this thing up in the next couple sets of notes. So I hope you have a good day. We'll talk to you on the flip side. So, hey, we'll see you. Bye.